In this module, I will discuss how microorganisms are classified. First, a microorganism is defined as a microscopic organism, which means the organism is not visible with the naked eye. These usually consist of a single cell or a cell cluster. These include viruses that are microscopic, although viruses are not strictly considered to be living. You will have heard of microorganisms as including protozoa, algae, and fungi. These types of microorganisms are eukaryotes. Eubacteria, which are known as bacteria, and archaeobacteria, which are known as archaea, are prokaryotes. And then viruses are also microorganisms, and these are not considered living because they cannot reproduce by themselves. The difference between a eukaryote and a prokaryote is that a eukaryote contains a nucleus and organelles, and a prokaryote does not contain a nucleus. I'd like for you to understand that there are three methods to classify organisms. The first method is to classify an organism based on its cell structure or its morphology. This is very simply classifying an organism according to how it looks, including its shape and its size. The second way to classify an organism is to look at its phylogeny, or its nucleic acid sequence. We can look at an organism's DNA to determine its family tree and classify it by that. This is referred to as phylogeny. The third way is to look at an organism's physiology or its metabolic capacity. This is how it generates energy, how it metabolizes. The first way to classify an organism is to look at its cell structure or its morphology. Cells can be classified as whether or not they have a nucleus, and therefore they would be either eukaryotes or prokaryotes. For eukaryotes, this includes all of the kingdoms, Animalia, Planta, and Protista, but the Protista are the only ones that are applicable for microorganisms. So algae, fungi, and protozoa are all Protista. A prokaryote does not include a nucleus, but has all of the properties of a cell, of a living cell, meaning prokaryotes can reproduce. A virus is not considered a cell, but is a microorganism. In this figure, you see the relative sizes of microorganisms. Eukaryotes are extremely large compared to prokaryotes, which are extremely large compared to viruses. I do ask for you to learn the average size of each of these. This is a figure from your book, and you'll see that size is issued here in microns. And you'll see many of the things we talked about that aren't microbial, such as suspended particles, colloids, flocculated particles. But up here, you'll see microbials, or microorganisms. Bacteria are considered to have an average size of around 1 micron, but this can vary, as would a bell curve. Algae, or eukaryotes, have a much larger range of sizes, but they are typically greater than 1 micron. And viruses have sizes ranging in the nanometer range. Fungi, Giardia, and Cryptosporidia are all other types of eukaryotes. And you can see here that all of these have fairly large sizes compared to bacteria and viruses. The second way to classify organisms is to look at the phylogeny, or the DNA sequence. Although most genes are very similar between all living organisms, there are differences that can help us classify. DNA sequencing began in 1975, and when you look at DNA sequencing, you can establish family trees, such as the one shown here. In this family tree, you'll see that prokaryotes, bacteria, and archaea have a huge amount of diversity compared to the eukaryotes. Relatively speaking, the DNA sequence of animals, which includes you and me, and Giardia, we are phylogenetically very similar compared to the diversity within the prokaryotic world, according to our DNA sequences. The third type of classification is physiology. This is also termed metabolic classification. I would say that classifying microorganisms based on size and physiology are the most useful for engineers. If we're going to physically filter a cell, we need to know its size. If we want to use cells to do work for us, we need to understand how they generate energy for growth, how they obtain carbon, and whether or not they need oxygen to live. In this course, I'd like for you to learn some basic terminology of how an organism gets its energy. An organism either gets its energy from chemicals or from the sun. If an organism obtains its chemicals from energy, it is called a chemotroph. If it gets its energy from the sun, it's called a phototroph. Within the chemotrophs, those chemicals can either be organic, meaning they have some carbon, hydrogen, and oxygen content, or they can be inorganic. Ammonia is a very common inorganic compound. If it gets its energy from chemicals, and it gets its carbon from organic chemicals, it's called a chemoorganotroph. If it gets its energy from chemicals, but not organic chemicals, it's a chemolithotroph. There's a second term here I'd like to show you, and that is that anything that gets its energy from organic chemicals is also referred to as a heterotroph. Anything that gets its energy from inorganic chemicals can be defined, and these would include both phototrophs and chemolithotrophs, as autotrophs. And this distinction here is really inorganic carbon versus organic carbon. So we have a lot of different names to talk about microorganisms. Heterotroph and autotroph is actually a very common way to discuss organisms because the difference of organic versus inorganic carbon is so critical. You can consider this in this course as just vocabulary to learn. Coming up again, just for a review, there are 3 methods to classify organisms: cell structure, phylogeny, and physiology. Again, cell structure or size is very important for filtration applications, and physiology is very important for biological treatment applications. Thank you.